I guess we're going to be kind of switching gears a little bit, starting with today's seminar, just because up until now we've been doing sort of a lot of broader conceptual project organization sort of topics like data analysis and project management and mentoring, stuff like that. But starting today, for the next couple weeks and sort of for the middle chunk of the semester, it's going to be more, I don't want to say technical because it's not going to get super in-depth technical, but more sort of practical skills and techniques that you can use when you're working in the lab or in the field, wherever you happen to work. So things you might have heard about, like conceptually in class or in your lab, you might have actually used before, but don't really know the theory behind them, why they work the way they do. Maybe something you read about in a paper once and you were wondering how it worked. So that's sort of the idea behind what we're going to be doing the next several weeks. In no particular order, we're starting with spectrophotometry, what I'm calling spectrophotometry and you. Because, whoops, I have my notes here. Yeah, so not only what spectrophotometry is, but how you can actually make it work for you, how it might be useful to you in your research life. If you've heard the word before, it was probably in general chemistry, if you've taken general chemistry, but probably not since then. If you haven't heard it before, then you'll have no idea what I'm talking about until I explain what it is. But kind of a big long word, but if you break it up into its component parts, really what it means is measuring light spectra. So making measurements of the spectrum of light or of electromagnetic radiation in general, using that using how light is absorbed or reflected or transmitted through a material or a substance in order to measure something, some property of that material or substance. So I tend to get kind of minimalist with my slides, so there's not a lot of words. It's mostly pictures, but I'm not sure how well you're gonna be able to see this. It's a little bit light in here, but wrong computer. So kind of a, I guess, review or crash course if you've never learned this before about the electromagnetic spectrum. So I don't want to get like super deep into the physics of it because first of all, I don't really know much of the physics of it. And there's not time, it's not important, but suffice it to say, electromagnetic radiation is a form of energy that's found as waves. Depending on the distance between the peaks of those waves, what we know is the wavelength, that affects the energy of the particular radiation. So the shorter the wavelength, the higher energy the radiation will be. Longer wavelength is lower energy. Longer wavelengths on the order of meters is your radio waves, slightly shorter is microwaves. We get into infrared. The very short wavelengths are the high energy ones, or the gamma rays, X-rays, and the UV. As humans, we consider the shorter wavelength energy, the higher energy waves, more dangerous to us because they can penetrate skin, they can do damage, which is why, you know, you have to wear a lead apron when you're getting X-rays, but not when you're listening to the radio. So one particular range of wavelengths between about 400 and 750 is the visible light range. And so different wavelengths represent different colors of light depending on what's being absorbed or reflected. I'll do that in a second. But when you pass, again, light or radiation through a sample, in this case we're going to be talking about a liquid sample or something dissolved in water, so an aqueous sample. If there's something in that sample that can absorb light, all of it or a particular wavelength of light, the light that goes in is not going to make it out the other side, so it's being absorbed. The light that does come out the other side is called transmitted light. Absorbed light is the light that is absorbed, that stays within the sample. You can imagine these are inversely proportional values, so transmittance and absorbance. You can't just add one to the other and get the total light you put in. It's a little bit more complicated than that, but regardless, the more light that's absorbed, the less is going to be transmitted, and vice versa. But different chemical species in solution are going to absorb or transmit light differently. And this is where you can start to kind of relate colors of things to what kind of wavelengths of light they are absorbing or transmitting or reflecting. So if you see something that's a blue color, what that means is that it's reflecting the light at the blue wavelengths rather than absorbing it. So it's actually tends to be absorbing kind of, based on this wheel, roughly opposite wavelengths to the blue ones. So this isn't perfect, but as a general rule, if it's showing up as blue, then it's absorbing nearly opposite on that color wheel to the blue wavelengths. So something that's white would be reflecting all wavelengths, because white is the combination of all colors of light. Something that's black would be absorbing all the wavelengths, reflecting none of them, because black is the absence of color. So also probably somewhat intuitive, the fact that the more concentrated a solution is that has a species in it that can absorb light, the more light it will absorb. So the more of the light absorbing species is in there, the more light will be absorbed. But that's sort of the key. The key behind how this whole technique works is that the higher the concentration of something, the more light it's going to be absorbing. So if you remember this one, Beer's law, which is how we relate this mathematically, the absorbance to the concentration of something. So absorbance, A, is related to concentration, which is C, by relating it to the molar extinction coefficient, also called the molar absorptivity, but this is a constant that's a property of a chemical species. The path length, which is the distance that the light is traveling through, and then the concentration of whatever the species is in solution. So every given chemical or chemical species or whatever you're talking about has a distinct molar extinction coefficient. Most of the time, you're not going to know what that is for whatever chemical it is you're looking for. So the absorbance, the units are dimensionless. They're just given as absorbance units. So if you see something given as, for example, A450, that's the absorbance when it's exposed or radiated with 450 nanometer light, because whatever is in solution absorbs at 450 nanometers, and so A450 is the dimensionless units for that. So the way you probably learn this in general chemistry, the way a lot of people learn it, is using the spectrophotometer with the little square cuvette and the square-shaped hole. Usually they're about a centimeter square because that makes the path length easy to deal with. But so you put concentrations of something, usually like food coloring or something, of different dilutions into the cuvette, measure the absorbance. You can either measure absorbance across all wavelength and see where it absorbs, where the maximum is, or you can choose one and measure the absorbance value at that wavelength. So they come in many different shapes and sizes. There are standalone ones, just sit on the bench top. There are ones that attach to a laptop, which are a little bit easier to use, but they all work the same way. So they're all passing light through your sample and detecting how much is transmitted, and based on that, figure out how much is being absorbed. But even though this is sort of the classic way to learn about it, probably you're not going to use this in a lab. I won't say never, because I'm sure some people do, but I haven't since general chemistry. But there are other ways that you might find the same kind of principle or the same technique in your real world life. So two ways. The first way is, I don't know if anybody's ever used one of these, the nanodrop. It's basically the same, it's the same principle as that spectrophotometer with the square-shaped cuvette, except it's tiny. You're using 1 microliter at a time of your sample, and it's actually super, super cool. And the only way I can explain it is to show you their website. But so this thing here, it's the Nanodrop 2000 UV vis spectrophotometer, so it means it can absorb can deal with UV wavelengths, it can deal with visible. So it tells you it has a wide spectral range from 190 to, that's tiny, 840 nanometers. But what this is, I mean, it's maybe like that big, sits on the bench top. There's a little tiny stage right here where you put 1 microliter of liquid onto there. And it looks like that. So that's the pipette tip going on there, putting a tiny drop of liquid and then it shuts. There's a kind of hinged thing here, and it makes it into a drop of a very known fixed distance. So it's basically like a tiny, tiny cuvette. So that's how it determines what the path length is. So then it will shine the light through, or the UV radiation, whatever it is, and use that as a way of quantitating whatever is in your sample. And most of the time, what I've seen it used for, and what a lot of people use it for, is DNA and RNA quantitation. So those nucleic acids will absorb UV radiation, usually in like 260, 280, so just out of the visible range. But what's nice about that is for nucleic acid, they do know what the molar extinction coefficient is. So you can actually just put a drop of your DNA or RNA right on this thing, have it measure the absorbance, and from that you can directly tell what the concentration of your DNA or RNA is. So it's super handy. If you don't know what the molar extinction coefficient of your sample is, which like I said is most of the time, then you usually have to do a standard curve. So you have to use solutions of known concentration of whatever it is you're measuring, compare your unknown sample to that, and use that as a way of determining what the concentration of your sample is. You can use that, you can use a nanodrop for that too. You'll just have to be putting drops on there over and over again with all your different standards. So it's not a huge deal. You don't use a ton of sample. Like I said, it's a microliter per sample that you're using. Yeah, so the other way, other than the nanodrop, which these are very common now, I know there's several of them on campus that I've used personally, but the other way you might see is using a plate reader. So has anybody here ever used a plate reader for anything using like a 96 well plate, like one of these? Awesome. So these are great because it's basically like having, 100 or so cuvettes at once in one little plastic piece. You can perform your assays directly in the plate, then put it in the plate reader to be analyzed, which is also very handy. But I kind of want to do an aside about the plate reader because, especially since nobody's used them, there are many, many, many different ways you can use them, many different types of plates, many different types of plate readers. So if you ever find yourself using one of these and never used one before, it can be good to go in kind of knowing what you're doing. So just like a little aside about plates. So 96 well plate is the most common type. That's these. They'll have 12 wells per row with 8 rows, so 12 by 8 is 96. They also make 384 wells, which are basically 4 wells for every one on the 96 well plate. They also make, and I've never seen one of these, but they make them 1536 well plates, so it's 16 for every one in the 96 well plate. So they're tiny. This was actually, this guy was doing this as an art project, like he made it a big mosaic mural of his face using microplates. So there were like, I don't know, a whole bunch of them, and they made an image. But yeah, most of the time you'll see the 96 wells, and they're very standard size. Like you're not going to have a 96 well plate that's any size that's different from this right here. And part of the reason that is a lot of people use kind of robotic equipment to go in and out of wells. Instead of doing things by hand, they'll use robots, liquid handling robots, and they have to be very, very particularly calibrated. And so you don't want plates that are all kinds of different sizes because that would mess things up quite a bit. But yeah, most of the time, 96 wells. But even within the 96 well plates, there's many, many, many options. So up here is just sort of a regular, ordinary, clear plastic one. They make them out of different types of plastic. There are types of plastic that can be frozen down to -80, so you can store things long-term at -80. There's just regular old cheapo ones like this. The ones I brought in here are super cheap, like bendable, not actually things you want to use for anything important. They also make opaque plates, so white and black. These white plates usually used for luminescence assays, so assays that are generating light. Black plates are usually for fluorescence assays, which require a special plate reader, and that's kind of its own topic. Fluorescence is very interesting, lots of things you can do with that. But so opaque plates usually used for something that's either generating light or you're using fluorescence. They can either come as one big chunk of plastic like this, or they come in strips, which sometimes they'll come in, you can break them off as strips of 12. Most of the time you can break them off in strips of 8, but that's really nice if you don't need to use a whole plate at the same time. You don't want to waste a plate, you only need to use half of it. And that's especially nice because sometimes if you're doing things like ELISAs, which you might hear about next week possibly, ELISA kits a lot of times come with a plate that's pre-coated with antibody. And antibody is expensive, and you don't want to just waste a whole plate that's coated with antibody. So if you can save it, use it next time. Just take the strips out, put them back in the fridge, and only use the ones you need. Sometimes you have, once again, especially for ELISA plates, you have plates that are coated with something, so they're high binding. They bind proteins better than just the regular, ordinary clear plastic ones. Some plates have like little filter papers in the bottom. Like there's a million different kinds. Different bottoms of the wells shape. You can have flat, you can have round, you can have V-shaped, which are usually good if you're trying to get every last drop out of the well. But the point is, if you're going, like I was doing last week, if you're going to Fisher or BWR and you're trying to find regular old clear plastic plates, it's not always super obvious and it can be a little bit overwhelming. And depending on how specialized your plates are, especially things like the opaque ones or the coated ones, they can get pretty expensive. Like more expensive than you would think for a piece of plastic, but once again, they're built to very specific dimensions and specifications, and so things like this get pretty pricey. So if you're in a lab and you go and use the wrong plate and you're using one that's expensive when you don't need to be using one that's expensive, you're going to probably make some people cranky. So good to know what you're getting into in terms of plates. So most of the time for using just a spectrophotometer based assay, where you're generating color and shining light through it in your plate reader, you're going to use something just like this, like a clear plastic one, but not flimsy, crappy clear plastic one. And the wells have flat bottoms. Opaque wouldn't work because you're trying to shine light through something. It has to be clear, but usually something like this will work pretty well. There's ones you can grow cells in the bottom of. Crazy. Varieties. So, kind of typical how a spectrophotometric assay in a plate goes. A lot of times, not always, they are kit-based. So you order this through a company and they'll send you all the different kind of reagents you need. Like I said, with ELISA, sometimes they'll send the plate with it. If it's not dependent on something that's coated in the bottom of a plate, you can just use a regular clear plastic one. Sometimes you have to supply your own, but they'll tell you in the protocol, use this type of plate for best results. So usually what you're testing for is some kind of particular analyte. So ELISAs for specific proteins. They make plate-based assays for all sorts of things, small molecules, lipids. I've used them for sort of phytochemicals, so chemicals from plants, finding out what the concentration of that is in a plant extract. But so many assays you can do in plates, but how they typically go. So like I said, you need a set of standards. If you don't know what the extinction coefficient of your sample is, of the color that you're generating is, which most of the time you don't. So you have to make up standards of known concentration of whatever it is you're trying to measure. Put those in the wells of the plate. You'll have unknowns. You'll have samples you don't know how much of whatever it is you're looking to try to measure is in your sample. So you'll add those to wells as well. And usually you want to do it either in duplicate or triplicate, just because sometimes you'll have a weird reading, and you want to make sure you're getting reliable results. So triplicate is great. A lot of times you end up doing it in duplicate because you don't want to waste sample. Waste wouldn't be wasting, but whatever reactants are involved in the assay you're doing that will generate color when they encounter whatever it is you're trying to measure. So you put those into all the wells, into the samples and the standards. Your color will develop however it happens. There's a lot of different ways it could happen, a lot of different reactions, based on the concentration of analyte. So usually what happens is you'll get a higher signal, more analyte you have. Sometimes ELISAs it's backwards, but most of the time you'll have more color developing if there's more of what you're trying to measure in the well. So then you read at an appropriate wavelength for whatever assay you're doing. So it's going to depend on what color you're generating, what wavelength you're going to read at. So like I was saying before, something that's blue in color is going to absorb most at the opposite wavelengths from blue. So you don't want to read at the blue wavelengths because you're not going to get any signal. So depending on what color is being generated, like this one's kind of reddish purple, you have to adjust what wavelength you set your plate reader at in order to get the proper reading. And usually, once again, it'll tell you in the protocol what wavelength to read at. And a lot of plate readers now you can just like choose, like I want to read at this wavelength. They used to have kind of filters where you can only use a couple. Nowadays you can pretty much choose any one you want. Create a standard curve based on the known concentrations you used and whatever absorbances you get, because now you will know that a given absorbance corresponds to a given concentration. You have a whole range of those, and you'll be able to use your standard curve to calculate the concentrations of your samples. So that's all I have for PowerPoint, but I do have kind of a hands-on example of that. I made up some, I only have a couple of them because one of them evaporated, but so plates with a standard curve I've made up and some unknown samples. And your task is to figure out what the concentrations of the samples are based on the standard curve. So you're going to be making a standard curve. I have graph paper. It shouldn't be that hard. It should be linear. But figure out what the concentrations of unknowns are based on the standard curve. And there's a couple of them on here that are going to demonstrate to you some things that are potentially confounding factors when you're doing a plate-based assay, some things that'll sometimes throw you off, that might make things a little bit difficult for you. So you should see those as you're working. So I have 3 of them, might have to share. Because like I said, this one's, yeah, had some issues, but give it a shot. Or I can hand them out either way if you don't feel like moving. And once you've done those, we can kind of regroup and discuss how it went. Yeah, or you can just, there's only part of it you'll need this for. It's just like your predictions about what the concentrations will be. You can, yeah. Or you can just use it up here if there's not going to be enough to go around. Yeah, for the beginning part, just because it's going to be like predict based on what it looks like, what you think the concentrations are going to be. But then the reading of it is on the back, and so you don't need to be able to see it to do that. You could, sure. I'm about to fall asleep.
You can do this. I know it's that time of day though. It's
Making me kind of hard to see, so put them against the white paper.
Give it my best shot.
Yeah.
Thank you, ma'am.